Okay, so our next presentation is from Augsburg.
Hello. Oh, okay. Hello, everybody. My name is Edwin Panora. I am a senior physicist at Augsburg University, and today I'll be talking about the characterization of swimming behavior of the dopamine magnum in the presence of a dopamine agonist and the effect this has on this living organism. So, our study aims to essentially create a granular level classification of the motion parameters of this Daphnia itself. So let's talk about what is a Daphnia. Well, a Daphnia is a microcrustacean aquatic animal or organism, and its nickname also known as a water flea, and it typically can be found in freshwater lakes or ponds. Its length scale ranges on a scale of 1.5 micrometers to up to 5 micrometers. Or millimeters, my bad, millimeters. And this is significantly smaller than that of its cousins, such as the crabs, lobsters, and shrimps. The physical makeup of the Daphnia essentially is one eye, two sets of antennas, and posterior spine. An interesting thing about this Daphnia is essentially while it is present in lakes and ponds, it's consumed by fishes. And in turn, this Daphnia consumes small objects in water, and this essentially creates a balance in the ecology of the freshwater itself. Also, while this Daphnia is present in fresh water, very harmful chemicals can be also present in the water itself. And Daphnia is used to analyze the ecotoxicology research into this Daphnia and the harmful chemicals in the water itself. So how does the Daphnia move? Essentially, the Daphnia is able to propel itself with its antennas. And this motion is called a power stroke. The cycle of this power stroke essentially starts by the antennas moving in a downward position, moving up until it hits the body of the Daphnia. And then at this point, it has reached maximum thrust and minimum surface area. Then it goes into this phase called a recovery stroke, as we can see in figure 3, where the antenna itself is retracted into its original position. At this point, the Daphnia can either go into another power stroke or it can just coast with time. So dopamine is associated, it impacts motion and on organisms itself, such as humans, worms, or Daphnias in this case. And also dopamine is a crucial element in Parkinson's disease due to the fact that the dopamine in humans is very low, so they don't have a high amount of dopamine that essentially causes stiffness and irregular motion. And what we are observing is this D2-like agonist, essentially that stimulates the dopamine receptors in Daphnias and essentially is used to restrict the motion itself. It impedes motion. So in looking into the motion of the Daphnia or in observing its free motion, we created this quasi 2D glass chamber. And behind this chamber, we have a light panel to illuminate behind the chamber. And then we have a point break Firefly camera aiming directly at it. And this is at a setting of 30 frames for over 40 seconds. And here you can see a free image of the Firefly camera and the Daphnia moving in its chamber. Now, to gather the position data for this free-swimming Daphnia, we use ImageJ and C-Track software to help gather the data itself. And here's tracking software working. Now, in gathering this position data, we were able to extract different motion profiles, such as the vertical, horizontal displacement, and the velocity itself. And as you can see, the peaks of the velocity essentially represent a power stroke occurring in a given time. At this point, we can see, at this specific point, we can see that a power stroke is occurring, and this is in relation with the hopping in the vertical displacements. At this point, between power strokes, we see that the Daphnia is essentially it's falling because of its decrease in vertical displacement. So essentially it's sinking. We observed different motions in during the power stroke and post power stroke phases. We classify these two different motions as hopping and zooming for during the power stroke. For the post power stroke, we observe motions such as sinking and drifting. And then a shared motion shared between the two is hovering, where essentially the daphnia is moving, but it's moving in place. These are essentially the criteria that help us characterize these different motions taking place. I won't spend too much time on them, but if you have any questions, just ask me after. And using the previous criteria, we were able to implement this using Python code, and you can see this taking effect. As for example, in our during power stroke for our hopping phase of the daphnia, we see that the red marks is shown in above the vertical displacement, or where the vertical displacement is greater than the horizontal displacement. And then for the zooming, we see that shown by the green marks, we see that this takes place when the horizontal displacement is greater than the vertical displacement. And then the center is the hopping or the hovering phase, where the daphnia isn't really moving much, and this is classified where the total displacement itself is less than 0.5 millimeters. Similar procedure is shown for the post power stroke, where the similar for the green marks represents drifting, sinking with the red marks, and blue for the hovering. From what we were able to gather, we saw that when we introduced this drug to the daphnia itself, we saw that its velocity and displacement per power stroke is decreased compared to its control data. Similarly, for both the power stroke and after power stroke, we saw that in observing the profile distribution of the characterization of the motions, we saw, and as you can see, the leftmost representing the controlled or undrugged daphnia, we saw that in introducing the drug itself, that the hovering motion has increased in the state of the drug, and also increasing in longer exposure times. From this idea, we saw that this is very interesting because overall, from the velocity and distance that we observed from the daphnia in a drug state, we inferred that while the daphnia is moving, it's not moving anywhere. Due to this idea, we wanted to look closely at the daphnia, specifically at the segments of the daphnia, the antenna segments of the daphnia. So we created this different set of experiments where we essentially have this chamber itself, this 14 millimeter chamber that is set on a pin and magnet to hold the daphnia in place in a belly down position. And we place this whole chamber under a microscope. Then from this, we have a photon camera recording at 1,000 frames per second for 17 seconds. And here is an image from that camera of this tethered daphnia. The daphnia has two different segments, the first segment and the second segment. And what we are specifically looking at is the change in angle between the first segment to the horizontal of the body. And then we are also looking at the angle between the first segment and second segment, represented as theta 2. From our observations already, we can already notice that while in a controlled state, the daphnia conducts a full power stroke, we see that the range of motion for a drug is more restricted. Thus, the daphnia is conducting a semi-power stroke. In averaging this data, we saw that for the first segment, or theta 1, we saw that the change in angle between the controlled and drug has decreased by 18 degrees for a drug daphnia. Okay, that's awkward, but yeah, I was missing a slide. I'm not sure why that's the case. But essentially from what we saw when we observed theta two, we saw that the drug had little to no effect on the drug daphnia itself. So theta two for both the control and drug, the plot itself overlaps, stating that the drug didn't have no effect on theta two, only on theta one. Yeah, that's so. From that, we can essentially infer that the second segment is a passive follower to the first segment of the daphnia. And this might be further proven when we take a dark field imaging where we stain those of the daphnia, and we saw that the muscle density connecting the first segment to the body, there was a higher muscle density than that of the hinge between the first and second segment of the daphnia. In conclusion, we were able to characterize different motions of the daphnia using different criteria. We saw that in the introduction of this D2 dopamine agonist, we saw that this daphnia behaved in a very unique way. And we saw that this impact of the drug on the first segment played a crucial role in the change of the behavior of the characterizations of the motions and the overall decrease in velocity and distance they travel per power stroke. I would like to thank my team, Dr. Romita Gupta, the armrest there, Francesca Sobio, and Dr. Beckman for this opportunity. Oh yeah, I'll take any questions.
Is there a reason you picked that particular species of daphnia, or did you look at other species as well?
Yeah, well, this, I'm not too sure about that, but because we wanted to see that they were very sensitive biomarkers, and due to this, we wanted to see the effects of low concentration drugs. So they were just really more affected in the drug that we were using.
Is there any effect of swimming in the out of plane? Like it's a aquarium, right? So like when they're swimming into the picture, out of the picture, that happen, or how that affect measurement?
So like towards the camera away.
Yeah.
We were just looking at it straight ahead in a 2D plane, but that is something that we were considering. We plan on implementing in like a 3D environment.
Right. Other camera.
Exactly.
Yeah.
So that's what we intend to further the research, kind of take care of that.
Did they ever get blurry?
Not that we saw.
Was it limited though? Did you prevent them from moving in that?
Yes, the chamber itself, we made it, we decreased this with significantly small, so it just traveled in that.
Oh, so it's a very thin layer.
Yep, my bad. And this is seen with the first.
It could hover though. It could flip.
Yep,
But not
it could.
swim in that direction very much.
Yep, exactly. Yep, and that width we gave it was around like 0.5 centimeters. So.
One kind of related, but did you test different actual individual samples? Like you have those two plots.
Mhm.
Did you apply the medication to multiples to get like a sense if there's variation?
Yes, we just took the overall of it. This was applied over like 25 different daphnias. Yep.
Great.
Good. Yep.
Piggybacking off of his first question, how do you think having a limited width on that has affected the test result?
Since we try to restrict that by minimizing the width itself, I don't know if I can speak on that behalf. I do understand that we are losing some data in that essence, but I think like I said, moving forward, just trying to see, observing what's going on in that area. We'll have to see in that next experiment.
I think it looks annoyed. But you're studying how it swims, not the fact that it's trying to get away.
Yep.
Okay, thanks again.
Be a nice screensaver.